Welcome, Jason Williams. How are you today?
Good. Thank you for the invitation, Carlos.
No, thank you for agreeing. We've uh, worked together, and you've taught me in workshops, and I thought this was a really fantastic opportunity for you to share with us and students how you got into this latest set of tools and use of Oxford Nanopore sequencing. So today we have the opportunity to chat with you a little and learn about your career and learn about graphs and analyzing this nanopore sequencing data. So tell us a little bit about your position, and we'll get started with that. How does that sound?
Sure. I guess I gave you a couple slides, so at least I'm not surprised what's up on the screen.
You tell me when to advance.
Yeah. That also requires me to remember whatever the next slide is, but I'll do my best. So yeah, I'm an assistant director, diversity research readiness at the DNA Learning Center, which is at Spring Harbor Laboratory. I don't know what that tells you very much, but let me try to break it down. I'll start from the bottom up. So Cold Spring Harbor Laboratory is a research institution that focuses on things like cancer, neuroscience, plant biology, where I got my start. In fact, actually, it happens to be my background picture today. So that's a little bit about the lab. And within the lab is the DNA Learning Center, where we focus on hands-on science education, including training for faculty, which is how I've often gotten a chance to work with you, Carlos. And because we both have the educational aspect, and we teach about genetics and genomics, but also that we work really in the context of a research lab, we often are really excited about bringing new things into the classroom. And nanopore sequencing just happens to be the latest thing. It's a favorite toy of mine. But of course, we want it to be more than a toy. We want it to be an educational tool that puts everyone, including students, in touch with the data that they're generating, but then also helps us to answer scientific questions. So that's maybe a little bit about how I gotten into Nanopore.
You're always trying to teach students about visualizing these big and often complex data sets. Tell us a little bit about what you think the power of visualizing data sets, including sequencing data sets, is.
Great. Yes, that's a good question. You're absolutely right. Visualization is extremely important to doing science. I'm going to broaden the term in just a moment with the next slide, but I better stay on this slide for a moment about this idea of communicating patterns. I really like this this animated slide, which shows basically a scatter plot, which I think you will have talked with your students about, pointing something on an x-axis and a y-axis. And the point of this animated slide that's on the screen currently is that you could have a group of data points, and they could all, if you average the mean on the x-axis or the mean on the y-axis, their standard deviation, their correlation, you could have different data sets which have exactly the same numbers. In this one, the mean is 0.5426, right? So to a decent amount of precision, they have the same. And then if you go out to the thousandth or the millionth or whatever, they change the number slightly. But the point is that you can get almost any pattern out of the data set by just looking at summary statistics. And in fact, the aim of this visualization is to show you can even make a dinosaur or a star or whatever kind of pattern. To our eyes, we can quite clearly see those patterns, but looking at the numbers just in a summary, we can't really see those patterns. And the reason why I also, I appreciate the word visualization, but I also make it to more the idea of how do you communicate patterns, is because visuals are not the only way that we can do that. And so for people who have low vision or no vision, that doesn't mean that you can't visualize. On my next slide, if you wouldn't mind advancing, Carlos, I also thought of just a quick analogy to this, which is a bit of a trick. What I have on the screen, what I have on the screen now is some music. I could like put you on the spot and ask you, Carlos, what is this piece of music?
Oh, you're bringing back piano lessons.
Well, I'm glad that you at least noticed that it's piano music, because it's we see the both hands, the bass and the treble clef. But this is even more tricky than what I'm asking you to do, to say what piece of music, because this is actually a piece, just a moment of one that I like, from Beethoven's Ninth Symphony. And it's a bit of a double trick, because the Beethoven's Ninth Piano is absolutely not for piano. Beethoven's Ninth Symphony is a symphony; it's not a piano piece. So this is even more tricky, because Liszt wrote transcriptions of all of the Beethoven symphonies for piano, and so you would not only have to know the music, but you'd have to know how the music sounds taken down from being a symphony to a piano. My point here is that the information is actually still there. If you were to play it on the piano, you would be able to hear Beethoven's Ninth Symphony and spot the patterns from the chords. The visual element might be a little bit of irrelevant here, but maybe the take-home message is that I bet for non-musicians, if you play their favorite, if you were to show them a copy of a song without maybe the words that they like, they might not be able to recognize a song that they listen to really frequently or enjoy if they just saw the music and they don't know how to read music. But hearing the song, they would be able to recognize it, even if it was played by somebody else, or if it was a piece that was without the words, or they changed it in some other way. As long as you kept the key pattern together, you'd be able to recognize it. And so no matter what our sensory input, there are ways to communicate patterns that come across, whether we're talking about literal vision with our eyes or with other types of sensory perception. So just a thought there to expand the idea of communicating patterns. Anything that helps us communicate patterns could fit into that idea of a graph. And in the case of music, we actually do have a graph. It has a y-axis, which represents the time, and it has different values that represent pitch. So it's not so different from our scatter plot, but it just gives a nod to the fact that we can use all of our senses to communicate patterns and to think about.
That's an awesome analogy, Jason. And I love the emphasis on communicating, and not just visual. It gets to this multiple means of representing and engaging with information, not just visual, but to communicate patterns.
Absolutely.
So what is this next slide that you shared with us? You shared a data set, and I often emphasize to students, garbage in, garbage out. The data we get, we have to, from sequencing runs, we have to assess for quality. So what is this quality control graph you're showing us?
Great. Great point, Carlos. Yeah. So I guess before we even talk about a visualization, a summarize of data, we definitely need to have expectations, things that we're looking for, and we have to understand, where do those expectations come from? Are they expectations about the type of experiment that we're running? Are they expectations about the biology that we think we expect to find when we do this experiment? What you're seeing here is a graph, and we'll look at them both quickly, and then we'll come back and focus on one. But what we have on the screen now is a line graph, and on one axis, the x-axis, we have a percentage, and on the y-axis we have a value. It's actually kind of binned, and I'll explain this. So let me first talk about what the data are and what we're trying to see here. So this is one of a half dozen reports or so that you can get inside of a software tool called FASTQ, or Fast Quality Control, that we use to look at sequence data. And this data set, since I happen to have on the screen a section called Summary Statistics, is about, I think, 250,000 individual DNA reads that we are summarizing in this data set. And what this data set is trying to ask the question is, how many of those reads are unique? In other words, if I were to look at 250,000 DNA sequence reads that I got from some organism, would I expect them to be the same or different? If I chop them up, in this case, it says the sequence length is 40 base pairs. So 40 base pairs, if you think about a password, if you had a password that was 40 characters long, it's probably going to be pretty unique, right? Hopefully, right. It would differ from another. It would be pretty hard to crack, even, with a computer to try to guess your password if you had 40. Well, what we hope to see on a graph, usually like this one, is that most of the sequences are unique. And if you look at this graph, on the x-axis, it tells you how many copies of the sequence do we have. And it's almost 100% of the sequences of the total percentage is only having one, somewhere between 1 and 2 copies. So in other words, most of the sequences are unique. However, there is a little bump. As you start going down the y-axis, it starts becoming a bit, I guess, logarithmic, where you have, well, how much of the sequence do you have greater than 10 copies, greater than 50, greater than 100, greater than 500, greater than 1,000, so on and so forth. So there is a little bit of a bump somewhere around some sequences that have a little bit more. There's a little small population, just by eye, maybe it's somewhere around 3 to 5% of sequences where you have more than 10 copies. So why is that? Well, your genome, as you may know, is, or any genome, usually has some areas of it that are pretty repetitive, where you have the same thing written over and over and over again. Transposons do this, areas of the centromere or the telomere where you have the same sequence motif repeated over and over and over again. So we'd actually expect that the sequence isn't totally unique, but that we'd have a lot of unique sequence data. And if we notice, those green check marks are there to say that, oh, this is good data according to whoever made the rules, the people who wrote the software, for what is good. If you go to the next slide, however, this is data, and I've just shown you all of the different reports, but this is supposed to be an example of bad data. Well, it at least has a warning. And so in this case, we see a line graph, and it's talking about the overall percentage of total sequences that have duplications. And here, you're somewhere in the 65%, I'd say, with that blue line, are unique, only one copy. But then there are a large number of sequences, sort of around the category that's called greater than 1,000 copies. So somewhere around, I would say, 18 to 20%. I don't know if it looks around there just by eye, where there's a large number of sequences that are greater than 1,000 copies. So it's given a warning here, but how would you know? Maybe it's bad sequence, something went wrong, but more likely it's a different type of experiment. So in a sequence experiment where we're looking at RNA-seq data, we're actually looking for transcripts of things that are being made in the cell. And actually, the cell needs to make lots of the same proteins all the time. There's just proteins you're going to need to make every day, maybe even several times a day, in order to maintain the basic function, things in the cytoskeleton, other types of proteins. So if it depends on what type of experiment you're doing. If you were doing an RNA-seq experiment, you'd actually like to see a distribution where you are getting lots and lots of copies of the same thing over and over again, because that's what the cell is actively transcribing. Or it could be that you have a really, really repetitive genome. So lots of plant genomes, there's some that have, there are many plant genomes that have genomes that are much larger than human genomes. And a lot of the times those are transposons that are duplicated. So it's sort of in the eye of the beholder, but this is a nice clue. And it's something that would be really hard, I think. I can't think of any other way other than to do a line graph or a bar graph to kind of communicate to somebody how many of your reads in a sequencing run are the same, and to give you an appreciation of whether this is what you would expect because your genome is repetitive, or this is what you would expect because you did an RNA-seq experiment and you're expecting to get lots of copies of single genes being replicated. So it all is kind of not just taking in the data, but really trying to ask the question, is it what I would expect? Does it reflect the biology that I know?
What does this percentage mean?
So it says, so percentage of sequences remaining if deduplicated. So this is related to the blue line at that at the very left-hand side, where you see that it says we've got about 60 some odd % of sequences that are unique. So one of the tools that you could do, and there's reasons for it, is you could say, okay, look at all of my duplicated sequences, and anything that's there more than once, just get rid of it. So you could normalize the data by saying, I just want to get all of the unique reads. And there are reasons why you might want to do that. If you were doing RNA-seq, you probably don't want to do that because you just gotten rid of all of your data. RNA-seq is sometimes a way of referring to it as a digital expression or digital count of expression. In other words, you're counting, this read has 20 copies of this RNA, therefore probably it's replicating or it's being transcribed at that rate. But another gene might have 2,000 copies of that transcript. Therefore, probably more of that is being made at any given time, or the transcript is more stable, or other reasons, but that's information. On the other hand, recently, in fact, actually I'm doing this right now, I want to know what the size of my genome is. And so in order to know what the size of the genome is, I actually don't want to get that repetitive sequence so much. I actually want to get rid of it because I'm actually asking the question, if I were to look at the genome, I don't know how big it is, and this is the case we're sequencing with some students, an endangered species, and we don't know what the size of the genome is. We have a guess based on other things, but we don't know. So we could ask the question, how many unique segments of DNA, if we break the DNA into 40 base pairs or whatever the length is, how many of those unique? Let me give you an analogy because it's kind of abstract. If I were to ask you what's the size of the English language? That could be a question. And let's say I'm not going to give you a dictionary, so you can't use a dictionary. I could have you read newspapers and then just basically count every time you find a new word and give you lots of newspapers. Let's say read every single newspaper for the next year, and every time you find a new word, right? Finding the word "the" is not going to be really interesting for you because it's going to be repeated many, many times. You do want to count it, but you only want to count it once, and then it's no longer new and you move on. Eventually, if you had a big enough sample, you'd be able to guess at what the size of the English language is based upon something like a newspaper. Obviously, there are lots of words that probably wouldn't make it into a newspaper because it's probably written at a certain grade level, but eventually you'd have some kind of estimate. So if we got rid of all of our duplicates, it's saying that we would have, that would be 70% roughly of the, in this case, data set 395,000. That's the percent of that 395,000 total sequences that we would have left roughly if we got rid of the
Really good
duplication.
point. A really nice way to think about it. It's to search for unique words in this case, unique sequences in a data set.
Mm-hmm.
You also shared, or I shared, this data set that I often ask students.
Mm-hmm.
Here's a data set from Nanopore sequencing, this technology where we have nanopores and we string DNA through these pore proteins and are able to now interpret those changes in current and call them bases.
Yes.
And this is one of the graphs that we often get and share with students, but it's rich in data. What do you think about this one?
All right. So yes, definitely. I love looking at graphs like that. And one of the things that you also, I think, pay attention into a graph like this is its shape. That we see this sort of big curve at the beginning, and then we see it sort of ski slope off, right?
Yeah.
So I guess maybe if I was a statistician, I could say this a bit more elegantly, but we might be thinking of other things, distributions that we might have seen in basic stats class, like the normal distribution, right? Where there's certain things that are average and they end up in the middle, and there's certain things that are really exceptional, and so they don't happen very often. Like if I ask you, how many times have you seen a person that is 5'7"? You probably see people who are 5 feet 7 inches every single day. If I ask you how often do you see an adult that's around 3'5", maybe once in your life, how often have you seen an adult that's 8'8", or whatever the extraordinary height is? And it's also probably pretty rare. So average things are average, and other things are exceptional we don't see very often. And so in nanopore sequencing, what I think we're seeing from this is that we're asking the question, if we ask how long a piece of DNA is that we sample with nanopore, we're asking how many times do we see a piece that's any given length long. And so there's definitely an average somewhere, if this is in kilobases, which it is, somewhere around 4 to 6 kilobases is average, what we're seeing here. The N50, which is a number that's an interesting number in and of itself. If we have time, I can explain it, because it's not an average; it's a little bit of a trick.
Right.
It's basically saying that we have an average number of sequences, and most of them are around 5 kilobases. What does that mean? Well, when we prepare DNA for nanopore sequencing, in many cases, not every case, but in most cases, we're trying to get the longest possible DNA reads, but DNA can actually break when you are pipetting, when you're moving it around. And so the more careful you are in the type of DNA extraction that you do, the more likely you're to get long reads. And oftentimes we want long reads because rather imagine a puzzle that had 20,000 pieces versus a puzzle that had 6 pieces. The one with 6 pieces is really easy to figure out, a jigsaw puzzle. The one with 20,000 pieces, it's a lot of work to figure it together. And so bigger is better in this case, but it reflects that there's some sort of cutoff. So if we look at the pieces of DNA there that are really short, you're getting some that are less than 1,000, like 0.13, which is probably as short as you can get a read, because any shorter than that, then the adapter sequences, there's nothing really that the DNA can, the adapters that are required to get the DNA through the pore. And I know you might have shared some visualizations or some explanations. It's not just the raw piece of DNA that we get in our tube, it's actually that DNA plus some other DNA adapters that we add to it. So there's a lower limit. In other words, it can't be shorter than a certain size. So we see a hump there where we see that the graph doesn't go off to the left infinitely. Then we see the average, and then longer pieces are increasingly rare. And oftentimes when you do an experiment, one of your goals is to push this curve off to the right as much as you could so that you can get longer, and you want the average to be in the above 10 kilobases, or the above 20 is really good.
Yeah.
So, yeah.
This was actually a genomics, a metagenomics sample, so a microbial community, and we got the DNA from this compost sample by beating the soil sample with beads for a while. So we shared some of the DNA. One other graph that pops up, and students and researchers in the lab often have questions about, is this mystery. How reliable are these sequences? And we often get stuck on this graph from nanopore sequencing. What do you think about it?
Okay, so let's take a look and see what we're seeing. The first thing that we're seeing is a Q, or quality score. And it does mention that this is a Fred scale. So you got to know all of a sudden, like, what are we talking about here? So what we're talking about here, there's something called the Fred scale, which is logarithmic. And basically it says, what's the chance that any given base in a DNA read is an error? So I don't have the chart in front of me, but if you get a score of 10, I think it's something like you've got a 90% accuracy rating. If you get a score of 20, then you're up to sort of 99%, and then 99.9%, and so on and so forth. It's an interesting thing because, sometimes you might think, 95 or 99% accuracy sounds pretty good. I'd love a 99 on a test. But then when you consider how much sequence you're often looking at, maybe millions or billions of base pairs, I think there's a graph or a chart at least out there somewhere called if 99% were good enough. And then it tells how many babies would go home with the wrong parents every single day, how many pieces of mail would be delivered. When you start to get really, really large numbers, the decimal points matter in terms of being 99.999. So here we've got a quality score and a cutoff, where I guess they're sort of making the intimation, if I recall, when you do base calling on Nanopore, which is actually getting better than this graph reflects, but when you do a base quality score on Nanopore, if you get below an 8, so now we're below 90% accuracy, it doesn't even register. It won't really call that as worth even looking at. The way that Nanopore often works is that if you're getting above an 8, and you're usually hopefully somewhere closer, like I said, now in the 20s, that means that, any given base might, any given read might have some errors in it, but if you get enough of the reads, you can average them out. So this is a way of showing the distribution. I guess it's saying that the mode, which is giving us the most frequent quality score here, looks like it's a little bit above 12. But the other thing that's kind of interesting here, if I see the bottom right, is on the time. So in other words, maybe this is the time of running the experiment. I think it might be in minutes. And I think what it's sort of suggesting is that as you run the experiment longer and longer and longer, the quality starts to dip off. And if that's what I'm seeing, the part of the explanation for that, I don't know the context of this particular experiment, but part of the explanation of that is that as you run longer and longer, there's actually enzymes and proteins that pull the nanopore, the DNA through the nanopore. And those start to either need more refueling. You have to actually add ATP, add fuel, or they can become clogged or other things to happen. They're biological molecules, so they can actually degrade. So I think what we're seeing here is that we get an average quality score. Sometimes it's a little bit higher or lower, which is the blue, which is what you'd expect. And then as you go on in time, at a certain point, the quality may degrade. And then that's, if you were running an experiment at that 60-minute mark, probably you would want to stop the experiment and refresh it or do other things to try to see if you can at least keep the average back up.
Yeah, this was actually a long run part of the previous data set. And this was, I think we did three days. So this is in the hours, and we noticed this large drop
that makes a lot
at
of
the
sense.
end, and people were concerned. But your point about this having enzymes and needing fuel is really an important one to keep in mind, because after a while the system loses energy basically to continue sequencing.
Yeah, you'll lose energy. The nanopores themselves can be clogged, or they can stop functioning because they are biological molecules. So it's like running at a certain point, you're gonna get tired.
You get tired. Oh, I like that analogy. And similar to that, we have another graph, the same data set, and now you can see the y-axis.
Yeah. Oh, hours
and the x-axis, hours and minutes. This one was similar, but it's the speed.
Correct. And it's quite related to the other graph. There's something called the translocation speed, which is how fast, how many bases per second are moving through the nanopore. And so this again is a reflection of it's a little bit tough to call it biology because we're not talking about a living system, but we are talking about biomolecules, proteins. So that is what's happening here as well. It turns out that in many cases, actually, that one of the technologies, and I'm not the expert here, but one of the things that the Nanopore company and the developers of the technology had to deal with was actually making sure the DNA doesn't go through too fast, because if the DNA actually goes through the nanopore too fast, it would make more mistakes because it's going through faster than it can actually be read. Of course, the faster you can get the DNA through, the more DNA you could read in a given time. So you get more nucleotides called, but you kind of have that trade-off.
Yeah, so, nice balance.
Yeah. So it's a balance, and they've gone back and forth, and they actually, when they design, I think there's another graph, maybe you'll have to look it up, but it definitely exists. I know I've seen it on the YouTube Nanopore channel, where they actually perform, I guess you would call directed evolution combined with machine learning to try to come up with new pore shapes, and ask the question, with this pore shape, what happens on an XY plot? Like one pore shape or one configuration of molecules might increase the speed but decrease the quality. And another one might be decreasing the speed and therefore the throughput, and the quality goes up. And the question is, where's the sweet spot that you can have both a lot of nucleotides and a lot of high quality? But this is reflected in the biology, although I think you see that the translocation speed doesn't change as much, or the drop-off doesn't look as significant, although my eyes, I don't know if it's really comparable, because we're not looking at a log scale on the left-hand side. Right. But I think that same time point, we see that the speed starts to change. It looks like it's going down. And then that's associated with the quality changing. Again, probably because things are breaking down and not working.
And along those lines, I noticed that Nanopore keeps on improving the chemistry, and between version 12 and version 14, I believe they changed the translocation speed and the temperature of the reaction.
Yes, the temperature, things that you all know from your basic biology, temperature, ATP, enzymes, right? The pH, all of those different things they toy with and tinker with. And you could see that happening in the graphs. And actually, although many people, myself included, don't always look at these graphs, they're actually things that you should be looking at while you're sequencing, and you can actually intervene to actually recover the sample and put a new flow cell, add some more fuel, mix,
maybe
etc. So yeah, those are all things, or really know when it's time to quit, because it's not going to improve no matter what you do. So all of those types of things are reasons why we want to pay attention to the graphs. And I'll comment, although we don't have an animation here, that many of these are animated in real time. So actually, as you're doing experiment, you can be looking at the graphs to make a decision about, do you need to do anything?
And what kinds of scientific questions interest you the most, Jason?
That's a very deep question. I don't know if I've thought about it. But I just
with Nanopore, maybe.
Okay, there we go. We can we can lower it to narrow it down to something. Let's see. So I think it is just absolutely cool to be able to sequence things real time, to be able in a few minutes going from some sample of interest to actually seeing the DNA sequence. So I'm really interested in making that process. I'm interested a lot actually in the biochemistry, in the lab side of it, of making that optimal. But I also love the data side too, of how can you help people to visualize data sets, and how can you help people to understand what's in the data and what the data could be used for, because I may have one question, but somebody else may be able to use that same data set to look at other things. So yeah, there's a lot that interests me, and I just love new toys. I have a kitchen full of toys. Every time something's out, I have to restrain myself. But I'm the same. Yeah, I like questions that involve new ways of looking at things. Let's say it that way.
Oh, I like that. How and when did you know you wanted to be a scientist?
I don't know. I know I've always loved science since sort of earliest age. So I guess I've always been interested in science. One of my earliest science memories, I think I could say, was a pop-up book. and it was a pop-up book about weather. And I do remember my favorite page in the pop-up book was a 3D pop-up hurricane, where you would see like the walls of the hurricane, and you could like twist it, and you could see how the eye rotates, and like you got the different layers of clouds and stuff like that. So I've always been interested in science, and I guess that's a form of visualization too, right? Being in a hurricane, it's just going to be wet and windy and complex, and you're probably worried about other things, but being able to appreciate it in that case through 3D, right? And something you could see and touch, that was always interesting.
That's really interesting that you love the 3D representation and also tactile.
Yeah.
And there's some math and modeling there too.
Correct.
So that may align with what led you to your current job. How did your pop-up book and hurricane lead you to this position?
Good question. I don't know. I just obviously took, nothing is planned. I took lots of science, anytime I could take science courses or be parts of science clubs or science competitions. It was always something that I was interested in. Probably the most influential thing, I guess I would say, and again, not being planned, is, from the moment I was in an undergraduate situation, I worked in a lab. Like I was like, oh, I want to work in a lab. My idea was in the science building I went to at Stony Brook University, there were 5 floors. And I thought to myself, well, I'll work on one floor every single year, like start from the top and work my way down. I never left the 5th floor, which was ecology and evolution. So I didn't quite follow with that plan, but I just always worked in a lab. And so that made it, when I came to Cold Spring Harbor, and naturally like, oh, I want to continue working in labs. I've been here for a little over 18 years, so I also kind of stay in places and just sort of sit and do science, and it's a great place to do science. So just being in the lab and being in the middle of whatever it is, obviously you take classes and you do things, you have to go on interviews at some point or talk to people. But I think, I didn't need anybody to tell me this is what I was doing. I almost felt it from the beginning, like, oh yeah, of course I'm going to be doing this. So
that's awesome. I did not realize it was 18 years.
Yes, Carlos.
Wow, cool. Here's a tough question. Okay. What is it like being a scientist who identifies as how would you identify yourself as a scientist?
Weird. So yeah, that was my answer.
Weird.
Weird. Yeah. I definitely always enjoy, humor, always enjoy a mix of things, whether it's music and science and art. I love
how in your workshops and webinars, you always bring in really great connections like art and food and I've seen you do this with students and also with like a webinar with faculty and instructors or computer scientists. So I love how you bring in different things. So what is a typical day in the life of Jason?
I also wish I knew the answer to that question. It would help me a lot. A lot of it is really talking to people like you, Carlos, which is great. A lot of it is a lot of the work that I do is people work. Making connections, like you said, whether it's conceptual connections, but also connections with people who are doing different projects. I've had the privilege at Cold Spring Harbor to really be able to teach and work with groups all over the country and all over the world. So oftentimes you realize what somebody is doing somewhere else is relevant to what somebody is doing in another corner. And so being able to think that way, it's very interesting because at the Learning Center, although we have lots of visitors, we don't have students like a university does. So it almost is a great combination because it means that everything we do is designed to go outward and to be communicated to people and to, but the fact that we don't have our own students that we just focus inwards, we're focused outwards. So a lot of my day is about interacting with groups and linking it to what we do at the Learning Center and hopefully linking them as well.
Oh, and I love how you you all produce and share resources and also make connections. So tell me about the moment when you felt like you really belonged in the field of science. Was it that pop-up book, or is it the day in Cold Spring Harbor?
Interesting. Yeah, I'm not sure. I know if, like, from a young age, I had a lab coat. I certainly went and just bought lab. Of course, I love wearing a lab coat, and I wear it now because then you can, even if your t-shirt, you've got to just throw a lab coat on and you're fine. You could sit in the room. That's a question that happens. That question or that identity happens many times, I would say, where you realize, oh, I get to do science for a living. So it happens, realizations. I think maybe one of the ones I remember, I don't remember where I was going, but sometimes sitting on a plane and going somewhere and realizing that people have invested their time and energy in having me come visit. And I'm like, oh, this is cool. And like people care, which is, rare and unusual. So I think that's certainly a moment that, and it's many, many moments of feeling like you belong in the sciences.
And those people are really lucky because you always do a great job getting us energized and interested in new topics. Oh, thank you for all you do, Jason.
Awesome. Thank you, Carlos, for all you do.